Okay. So the spinal cord is the way that your brain, up at the top, can command the rest of the body in this case. Like I said, this is blue text because I'm not going to strictly ask you, what are all these parts? Obviously, I'm not, but I just wanted you to get across, hey, if there's a command in the brain, it has to go through here, and it has to say like, okay, like we're going to go down this way and cause this contraction, or we're going to go down this way and cause this contraction down here. So these gigantic nerves, these are neurons, and they are going to be able to signal and say, hey, we need this area to contract, we need this area to fire. So, fun stuff that if you get into orthopedics, for example, you'll get to learn all of this. But for our purposes, this is still just a very good way to see kind of how this highway is regulated. You can see certain things sometimes don't work out the way they should. That's okay. There's treatment that can help scoliosis in this case. But the other thing that I want to show you, especially on this side right here, is how many of these nerve bundles are running around this column. You can't see it in the x-rays, but they're that dense because there's that many different highways that need to communicate with each other. So what this allows for us is we can use the spine and we can use the nerves that it's, kind of lending to communicate to the body. We can see how they're doing by testing and how the spine's doing by kind of testing, in this case, your reflexes in a lot of cases. So that's why for any of you that have ever injured your spine or your back, and this person, they're 1, 2, 3, 4, their C5, as you can tell right here, is fractured, unfortunately. If any of you have ever broken your spine, you know that that is a very, very bad time because you can see all these nerves right here. All of them are really angry at that point if you're, if you're causing them pain in that case. Okay. Now the good thing with spinal health is this is something that you can work on. This is the idea of standing, sitting less, going on walks, things like that. Regulating this whole column and making sure that it is very much used to activity is a very good thing. This is something you can tell your clients, your patients, anything. That's kind of the benefit behind this low impact stuff, that sometimes it's like, hey, I'm not like working up a sweat or anything. What's the benefit here? And it's usually this. Because if this highway ever gets disturbed, that's when you start to allow bigger problems to crop up, essentially. Okay. What are we doing when we take a look at reflex? So this is red text, but I don't need you to know, every piece of this. But the reason that we actually, poke and prod people at the doctor's office is because we're testing all that stuff from lab 1. We're testing that system from lab 1 to make sure that everything is firing as it should. So if we have some sort of stimulus on the skin, and God forbid it's a needle, it should just be one of these hammers. That would suck, that would hurt. So imagine the hammer, not the needle right there, but any sort of stimulus is going to show up here. The immediate effect of that stimulus is going to be read by nerves at the skin. They are going to go into the spine. Remember, there's our friend afferent, so it's on its way to be processed. So you don't need to know like the details here, like these little things. You just need to know that this signal, when it's felt on the skin by those cells, it has to find its way through the spinal column. And what happens is, if in the right spots, which medicine has identified certain spots that are supposed to trigger this reaction, what this is going to show us is this stimulus being coordinated and translated into the efferent pathway to a new nerve that in this case is going to go and react by stimulating a muscle. So this is the complicated way that when you get your knee hit by a little hammer, what that's testing is your spinal column specifically. Is this whole area right here, is that all functional? Is that smooth? It's very much not to see, is your knee okay in any case? What that's doing is that certain spot right there, that reflex is a kind of a highway right there. If you get stimulated at the knee by getting hit, that signal has to travel through here in the spinal column to make a reaction in the muscle. So we're not necessarily looking to see how the muscle's doing. We're kind of using this as a test to examine, is the spine like crunched in the wrong way? Is it cut off for any reason? Because let's say you do actually stimulate this and you hit it and nothing happens, this may be blocked up for some reason, right? This is a lot cheaper than ordering a scan. Am I right? It's terrible. Some people don't like those machines either. So what reflexes allow us is a very cheap, quick, and effective way to see, is your spine okay? Are your motor neurons okay? How are we doing? And this will definitely be posted, obviously, but it's kind of an application of Lab 1. There are sensories that we get the stimulus from, and they're going to cause some sort of effect. Actually, let's do this. Okay. So let's actually imagine a patient, and we're just going to do a spinal column and obviously brain. So in the case of your spine, things flow downstream. And there are many sites that we can actually test for reflexes to see how far downstream something is wrong, let's say. So in a lot of cases, let's say we test kind of one of the arm ones up here. So there's a tricep, there's like an elbow reflex you can do. That's kind of higher, that's closer to the brain. If everything's fine, then we'll see even response. And keep this kind of noted right here, is that you're going to test reflexes on both sides, right? Doesn't necessarily mean you're unhealthy if there's no reflex, as long as there's no reflex on both sides. That means things are even. So let's assume this first reflex up top here, everything's fine. Now let's assume right here, one of the middle reflexes, let's assume that there's no sign here. So at this point, what that signifies is no action, no reflex was shown. There's not, something was wrong. So usually what happens next is that anything below, like I said, spine flows downstream. Anything below some sort of injury, which is what we're assuming kind of in the middle of the spinal cord, let's say. And by injury, I mean like a small thing. We'll get to bad stuff. If there's something upstream that's going to interfere, this could have a really large reflex on one side and a really small bad one on the other. So in that case, below injuries are often uneven reflex responses. At injuries usually means no response, so at the site, and everything above injuries is still going to be fine. Because so long as you can get up here to the brain, this highway right here is fine. It's everything that we're assuming is going wrong in the middle here that's causing trouble. So those are some rules here. I'll actually screenshot this probably. This isn't the worst thing to post on the slides. So you can find patient, you can kind of start pinpointing where a patient has some spinal health issues by finding spots that do not work and anything downstream that is now uneven or kind of messed up at this point. And like I said, this is way better than scanning. The scans cost a lot of money, and that jacks up insurance on people that don't have what it takes to, pay or something. So you don't want to do that to your patients. So although we can't do the reflexes specifically today, so that whole section about reflexes we're skipping, you're welcome to try one of the hammers and do the knee stuff and see like, hey, are you like even, are you not, things like that. And you can always give that a shot. We're just not doing the reflexes stuff today because you had the exam and it's easy enough to get rid of it here. This basic idea is on the exam though. Penny made that pretty clear for me. So there will be a test question that's like, hey, if you have an injury at this point, what would you expect downstream, above, and at this point, let's say, something like that. So that's kind of the point behind doing all those reflex stuff, is we can pinpoint areas in the spine that are not all the way there, all the way right. Something's wrong in this system, for example, and this is how we can pinpoint that. Okay. All right, kind of as proof of principle, but still applies directly to medicine, obviously, is let's talk about grievous or critical injuries to the spinal cord. And what I mean by that is the gray matter is severed or completely gone. This is a little easier to track. If you have severed the spinal cord, let's say down here at the lumbar, everything below that part of the spinal cord that is responsible for, to be commanded by motor neurons is no longer commandable. If you sever the gray matter, everything below it downstream is done. That's it. As you can tell, the further upstream any critical injury is going to be, the worse outlook the patient's going to have. So typically, you'll see something like this, and it's hard to tell sometimes if critical injuries are going to be permanent, because you can't 100% tell if gray matter can ever reform in some cases. Sometimes car accidents can cause this phenotype to kind of show up, but not stick sometimes. Sometimes the gray matter can reform through some other means. But in a lot of cases, if you have any severances at any point, the downstream of that is going to be very difficult to control at that point. So this is different though than small injuries, where that's for reflexes, that's just like, hey, like you sit down too much at work and your spine is kind of uneven at this point. This is just, oh no, you're go ahead.
So basically the higher up the injury in the spinal cord, the worse your paralysis will be, or whatever the side effect.
Yeah, and then paralysis is a good word, because you have no more voluntary control of all the systems that we're going to see today. If you've severed like below here, nothing's going to work the rest of the way down, because all the signals that the brain needs to send those commands, they all flow downstream. So everything above that injury is still good. So in this case, this person would still have their arms, torso, neck, things like that, but everything below there is now out of control, not contractible. Okay. All right, so given that, how do we move all this stuff? So again, revisiting a little bit of lab 1, humans have to do stuff. Living things have to do stuff. And the skeleton's heavy. You gotta move it around somehow. You gotta react quickly. Favorite picture, of course. Would be so fun if that was real. Well, not for that guy, but still. All right, so why don't we take like 30 seconds and we'll go through before we start here. So what we're gonna do essentially is see how muscles are commanded to, from a relaxed point to contract, basically. So obviously I'll post something on YouTube or from YouTube that is like a good reflection that you can follow along here. But I like to go over this too to make sure you have all the tools that you need. There's 3 main stages. First off, you just need to get a motor neuron, like the stuff that we just covered, that kind of flow throughout the spine. That needs to contact a muscle. When that contact is made, that muscle can be stimulated. That's the AP that you're seeing. Then the final stage is when a muscle is stimulated, it goes from a relaxed to a contracted state. In healthcare or exercise phys, you are going to come up against big pathways. This is one of the first ones. I think it's good because it is a pathway that you can, has a lot of checkpoints and a lot of reasons why it makes sense, I think. other pathways in human physiology can be a little more ornery, to say the least. Now, everybody's going to kind of change, and this is where things really start differing from anatomy in this class. Is pathway notes are very different than here's everything, right? This is a sequence. The first time I see a pathway, honestly, there's nothing wrong with just being like 1, 2, 3, 4, and just down what small stuff is happening each of those steps, okay? If you want to draw it out, the whole thing, like a big flow chart, that also helps. But whatever works, that's what you should kind of key on. Okay. So our goal is to go from right here. This is the end goal. This is all 3 stages. This is the whole thing that we need to get to. Most of your muscle fibers are in this relaxed, long state. We need to shorten that. Now, it doesn't seem like much, but you have thousands of these fibers working together in these coordinated actions. And like I said in anatomy, you learn all these muscles that they're all opposed to each other, right? Biceps versus triceps, quadriceps versus hamstring. All of those work together to make fluid motion. So in this case, here is a big muscle fiber, and here are these small sarcomeres, which are just little units of these things. So muscles made of muscle fibers are made of sarcomeres. Why don't I just write that down? We'll see a sarcomere in a sec. So we'll keep this in mind too, just because some of the stuff that we'll get to see in this case is that when you think about your muscles, and right now this is just green text, when you, let's say, lift your bicep a little bit, and it's like a 2-pound weight, you're not actually going to have to recruit quite as many of these fibers, right? Like what's the number, higher or lower? It's actually going to be a little lower. Similarly, that's the number that you're going to lift with the sarcomeres. Now, as you lift something bigger, like you try and lift a chair or something big, or like a desk, you will recruit closer to 100% of what your muscles can do. So that's kind of where we're going. But let's see how we get one to fire just on its own, kind of at this biochemical level. This is a sarcomere. This is that small unit, the unit that's made of these really tiny parts. And again, this is kind of the end game right here. This is the end stage that we want. We take a long fiber, and it can cause itself to contract and become short. And that's the whole idea. It's definitely going to help to know which pieces are myosin versus actin. We'll cover that, but obviously stuff like this is always going to be posted for that reason. All right, so let's get back up here. Let's look at motor neuron. How is this going to initiate that process? So first things first, let's imagine at least right here, like a big muscle cell or a big muscle fiber, okay? Now your muscle fibers are located throughout your body. Like we saw though, your nerves kind of splinter throughout your body too. So in a lot of cases, let's imagine this is like one of those big nerve axons. And what it can do is basically makes a little bit of contact, makes a little pieces of contact with muscle. So there are points in your muscle where they are physically interacting with your neurons, specifically your motor neurons, obviously. So starting from something familiar, if we have an action potential traveling down this neuron, right? Because that action potential had to come from the brain originally and say, trigger to fire that neuron downstream. Downstream, 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 until we get to this point. We're at the final stage of the neuron, and this is actually where we'll end up leaving. The signal becomes less of a nervous system issue, and now it will become a muscular one. So in this case, what the action potential is going to do is cause a big rush of sodium to show up. What this is going to do is basically cause a lot of, and this is a pretty fine detail, but again, you're going to see why this matters specifically with drugs. What the end game of this action potential is, this positive burst with the sodium, is to cause calcium to rush inside the cell. Calcium's only going to show up once that charge is there. And that's the purpose of the action potential, is to provide that positive burst. Calcium's kind of a third party here, right? We haven't really used it in the strict action potential way, but it does show up here in the muscles. First here. So, action potential finishes on the end. This is going to cause neurotransmitter to release. Now, our neurotransmitter in this case is going to get released from the neuron, and it's only going to do this if calcium shows up, which only did it because the positive sodiums bursted it through the action potential. So in blue here, something called acetylcholine is going to show up. Let's zoom in on what's going to happen down here. Okay, so I'm zoomed in now at this part right here that's kind of showering the muscle cell with this acetylcholine. Okay, so we'll call this fiber. Okay. So you can call it ACH for short. So this acetylcholine is a big chemical. And what it's going to do is move across from the neuron to the muscle. So the muscle cell stands ready with these channels. So these channels exist as kind of a gateway into the muscle cell. What acetylcholine is going to do, and the reason that once it's released, all this is going to trigger, is acetylcholine is going to go and it binds to these specific muscle transporters right here. Once it does that, the sodium that is waiting outside the cell, which again, remember, there's tons of sodium around. Once acetylcholine makes that contact, the sodium can rush in. Now, the interesting thing here is that acetylcholine can cause these little channels to open up and allow sodium in. But once a little bit of sodium gets in, it starts to turn on these receptors from basically from behind here. And then more sodium that was all waiting out here can also come in for this reason. So it's kind of twofold. First step, yeah, you need the acetylcholine to open up the floodgates. Once the floodgates are open, more floodgates open. What happens here is that, again, wherever sodium goes, there's so much of it, the charge is going to get positive. Okay. So this is a good stop. This is a good like checkpoint right here to just go back and see everything again. And I'll actually go ahead and do, I don't have any colors to do. Oh no. Give me a green. There we go. Oh, boo. Come on. There we go. I'm trying to scroll up on here, but it's not working. There we go. Okay. So that's kind of the first set of stages. So we made it. Essentially what we accomplished was there was a signal that was transmitted from the nervous system as that action potential, that positive charge. We translated that to an activation in the muscle cell. There's a lot of work for that, but now we have a muscle cell that's going to get activated. This is good. So first thing that happened, that action potential was coming down the motor neuron. I'd say second thing that happens is that positive charge from this action potential, all that release and that flood of sodium in, and we'll see sodium and calcium running around in different roles in this. Calcium at step number 2 is going to be allowed in. Step number 3, as a reaction to the calcium, acetylcholine in blue here is all released across that connection between the neuron and the muscle cell. Step 4, acetylcholine is going to bind all of these sodium channels on the muscle cell, not the neuron anymore. So this transfer of power is essentially kind of happening right here. Because acetylcholine opens those gates, sodium rushes in. I promise, I hate taking up time here, but I do promise this is on like the lecture, the lab exam, the next one. So that's why I want to give you guys all the resources here. So sodium floods in. I'd kind of call this like step 5.5, is that kind of another consequence of sodium flooding in is that more sodium floods in too. You can open the gate from behind too, as long as that charge is there. Okay. And then step 6 is essentially you have super positively charged the muscle cell now. The charge in the muscle cell was just kind of nothing, whatever. Now it is positive. Okay. All right. Going okay? Middle, down, up. Doing okay. All right. Don't worry. Like I said, like YouTube videos are a great way to follow along this, pause, things like that. And I am actually recording this one. So if it would help to see this section again, you can email me and I might post this. Okay. Go back to red. Now let's actually look at this system as though we are now inside the muscle cell itself. We've already done all of our work by supercharging and adding all this sodium. Hooray. Okay. So let's actually look at, I might use this little thing too. Sorry, yeah, and you can also play with this little thing, but I don't know, I don't think it's that great. So, okay. So the way that these fibers are formed is of these little sarcomere units. So let's look, kind of imagine them like these big bridges across here. And I'm actually going to move this one over here, sorry. Okay. That barely made a difference. I don't know why I did that. Okay. So what you have in these sarcomeres is, sorry. What you have in these sarcomeres is an overlap of 2 molecules, myosin and actin. I gotta add one more. Okay. So in your cases, or in the case of, in this one, I just want to make sure. I always mix up myosin and actin, so I just want to make sure you got the exact right thing. Give me a break. I know, how many of your professors like check their notes on stuff like this? Sorry. Okay. So what's ultimately going to happen here is that myosin has these little heads on it. And what they're doing when they're relaxed is that they are kind of bound to the actin units that are kind of around them, right? So in this sarcomere structure, they're kind of just sitting like this. The end goal of where we want to go with this thing is we want this to kind of contract to this size, right? So this is our ultimate goal, is to get here from where we're at up here. What these little heads do, these little combs basically, is that if given the chance, and if told to, they're kind of going to pull at these contact points, and they're going to pull this back. And they're going to do the same thing on the other side. That's the end goal here, is that you want to move these units using the myosin fiber right there. Now, they're not just going to do that, right? They need to be told to do that. So we'll cover a disease where your muscles do move involuntarily, and it's not a fun time. So normally, you do want your muscles just relaxed if you're not commanding them to move. So we actually have to zoom in on one of these right here to see a couple things. So typically, these two things are not going to be able to interact, because once they interact, the contraction happens and they pull each other in. You actually have two molecules that sit on your myosin and prevent it from binding to actin. They stop it. So right now, as you're sitting, and the muscles that you're using, that you're not using, they have two molecules preventing that interaction happening. There's one called troponin, and its partner is called tropomyosin. The entire role of these things in blue is to prevent myosin and actin from binding, because the minute they do, they're going to rip each other in and contract. I know this thing looks like a caterpillar. I try to make something that like has some sense and has two parts to it. Otherwise, the two can be a little weird. So both of these, they sound the same. They have the same goal: block this interaction, block the interaction between our myosin in red and our actin in black here. Because once those little combs on the red and actin interact, or on the red and myosin interact with the black actin right there, that's the contraction. They're going to rip them towards each other. And that's the muscle contracting, but just at a small level here, because we're only looking at one unit in that fiber that's made of maybe thousands of these things. Okay. Let's do, okay. So how do we get the troponin and the tropomyosin off so that the contraction does happen, because we have been commanded to, follow this or contract this muscle? So one of the things that sodium does, once it's inside and once this positive charge is here, it finds its way to something called a sarcoplasmic reticulum. This is basically just a little structure inside the cells. And all it does is holds a bunch of calcium in it. That's it. It's like a bank, and it's just got all these calciums in it. And they're trying to get out, but once they're put in there, they can't, because they have their own like membrane, and it's all sequestered right in there. Now there's a reason behind it being sequestered. The calcium is the reason that we're going to unlock the muscle. But once you have this sodium show up with all its positive charge, the positive charge is going to basically open up sarcoplasmic reticulum. It's going to allow everything to spill out. That means all these calciums are going to rip out of the cell, or rip out of the sarcoplasmic reticulum. Calcium is the key to getting rid of troponin and tropomyosin. So what's going to happen with all these calciums is they are very heavily attracted to troponin. They're going to go and they're going to bind it, and they're going to get rid of it. And once you get rid of troponin with your calcium, tropomyosin runs away too, because it doesn't have its binding partner anymore biochemically. But all you need to know is that once you get rid of one of these with calcium, you basically unlock the final stage of this process. You have a lot of safeguards not to contract, but what we're getting rid of here is all those safeguards to contract, because the body told us to, and that's the trigger and the importance of that sodium burst. So once these two are out of our way, myosin and actin are going to start interacting a bunch. Let's do a green right here. All these points of contact right here are now going to make that contact. And essentially, what happens once they're free of troponin and tropomyosin, they basically all, they're kind of like little, like, I don't know, like fingers kind of. Basically, they're just going to, I know that I'm like doing gravity here, they're basically just going to be like, we start right here, and they're gonna kind of just like grab at the actin right there and basically pull it piece by piece. So for example, we're looking right here, like this one can make contact right here, push it this way. That's gonna allow this one to make contact with it as it's moving, make contact, push, make contact, push. So each of these little beads is essentially going to grab, release, grab, release, grab, release across the board until you get the contraction down here, which is the actin is fully brought in from where it started on the outside. Let's go like that. Okay. Now the formations that cause the grab, release, that's actually that ATP molecule being used right there. ATP is the trigger to grab and then release over and over again, and you need one each time to make that happen. So we'll get to a slide that does that a lot cleaner than I can draw it. But starting at this stage, starting back from 6. So once we released calcium into the cell, calcium arrives and bursts out the sarcoplasmic reticulum. It allows that to open up. Step 8 is that all that calcium floods out. All that calcium for step 9, sorry, I'm running out of colors here. Step 9, the calcium is essentially going to get rid of, come on, the tropomyosin and the troponin, basically the things that say do not contract, because again, you want a lot of safeguards. You don't want it to be contracting all the time. And then the final stage is all of these points of contact between actin and myosin are actually going to fire. They're going to contract from both sides. And you end up with this small little unit contracting from the distance that it used to exist at to a smaller one. And that's how muscles essentially move. And they do this 1,000-fold, obviously. All right, so like I said, I promise that I won't be up here as much in the next few, but I've seen how many exam questions come just from this process. So it pays to have at least one repetition at it, take a few more with the videos. And like I said, kind of practicing it on your own, even if it is just numbering it, is a super good way to handle this. Okay. So let's actually look at, okay. So this is something that's mainly going to show up next week, mainly at least, but we do kind of see it today. But we're out of the pathway, so we did it. We contracted the muscle cell. What happens was happening at a scale that we saw just at one. We just saw one unit, and this is a very good way to see that scale applied. And this is why many and thousands of those little units are able to move stuff, do stuff, everything like that. So again, this is super important as far as, for example, just typical lifestyle, athletics, exercise for health, anything. Because if anything's inhibited about this process, you start to lose that ability. So this is kind of what I talked about a little bit here, which is if you have this proportion of nerve fibers that are saying, hey, fire, fire, for example, it's kind of like the neurons, again, where if If you have to lift something light, you're only going to recruit a few of your fibers. If you have to lift something heavy, you're going to recruit all of them, or 90% of them. Kind of a fun side thing to this is that it's very rare for humans, the human mind, to allow itself to actually lift 100%, because there's a much better chance if you're recruiting 100% of your fibers that something's going to rip. That's typically why only in an adrenaline surge or some sort of fight or flight situation will you ever actually have achieved 100% recruitment, because it's kind of a trade-off that it's kind of a dangerous thing. So we'll actually get to see this with the frog legs next week, but this week we'll actually measure it on ourselves with as you lift more weight, basically, you're going to have to recruit bigger numbers of fibers. And this is just the stimuli to the nerve is going to recruit more at the same time. Cool, okay. So again, we'll get to revisit this next week a little bit too. All right, so now it's just kind of some green tech stuff. You're probably good for the notes. That was a lot of dense stuff, and you did just take an exam today. I always feel bad that lab 4 is the exam one. It's kind of a hard one. So with something like ALS, this is also known as Lou Gehrig's disease, where you progressively lose control of your body as far as your motor function. For a lot of reasons, it's usually that this point of contact between the nerve and the muscle starts to fail. That is not always the nerve's fault. It's not always the muscle's fault. That's why this is a very difficult disease to treat. Similar to any variation of Parkinson's, things like that. There are a myriad of factors that we are investigating to say, how can we treat this? Because once you find, say, a source at patient 0 to say, you can start addressing not only how to treat it, but maybe how to never allow this to happen, for example. But the degeneration of these nerves is typically what happens. And that contact, that point where the exchange is being made of acetylcholine, that's not actually happening anymore. On that note, actually, let's skip up. I missed some, I forgot something. All right, one last note in this process. We can call this step 11, sorry. All this acetylcholine that's up here stimulating the muscle to contract, there is an enzyme which is just going to take the chemical and chew it into pieces called acetylcholinease. That's in your lab notebook. All it's going to do is be like, all right, we're done here. Got to get rid of that acetylcholine or it'll keep contracting. Sorry about that. I always forget that one. Because the contraction reaction has to stop eventually, right? You don't want it to just keep going after you tell your muscle to relax. Okay. So as far as PT, exercise, phys, OT, a lot of what you are relearning is at this junction that we just covered. Some of what you're relearning and retraining can sometimes be those sarcomeres and small fibers, but a lot of what you're doing is actually retraining the neurons that are interacting with the muscles, that this is the right, basically you're educating, kind of re-educating them that this is the right path. And that restoration is a mental and physical process. Like you have to build confidence in your patients that taking another step forward is not going to, cause them to fall over, for example. And that process is a long way. Brian Shazier for the Pittsburgh Steelers here is seen walking after a close to grievous spinal injury in this case. But with the right setup, sometimes you can, like I said, renew those really difficult injuries. And a lot of this is reteaching your body to recruit those fibers kind of at the neuron level in a lot of cases. Because if you injure that spinal cord, that's where some of that injury is going to show up. So in this case, this is kind of the applied system of that. Again, it's in blue text. You don't have to know any of this, but this is a lot of what PT and OT are doing, which is basically relearning these motor skills. And what your body is doing is gaining confidence again in itself. I had a family member get in a car crash when they were younger and had to do the whole thing where you have to relearn how to walk. And it's not like you don't know what walking is. Your body just doesn't remember because the nerves that used to exist and command those muscles, they got damaged. New ones are going to need to form and make those connections. And it takes a while, to say the least. Okay. I want to speed through this, but there's different levels of actin basically. So this is the difference between fast and slow twitch in a lot of cases. It's a gene called actin-3. Basically, if you have actin-3, you are a better sprinter in a lot of cases. But if you don't have actin-3, if you have the like bad version of the gene, quote unquote, your muscle cells have more room for mitochondria, which allows you to be a better distance person. More mitochondria, more energy, longer distance. Less mitochondria, more bigger fiber, faster sprint, but not going to last as long. Same thing here. I love this because these poor dogs. There's another gene called myostatin that if it is knocked out in both genes, you get what is called, sometimes called a bully whippet, and their muscles are just like overgrown like this. This dog doesn't train or anything. That's just how it grew. So humans that lack both copies of the myostatin 1 gene, they also can typically sometimes look like this, right? Like the training can have a much greater effect. You might ask, hey, like why didn't everybody evolve to have that? Well, evolving as Homo sapiens wasn't always a huge advantage if you had to feed that much muscle, right? Not as good of a thing, right? So that's why something like this only shows up once in a while, is both versions of the gene are gone. So let's look at a couple diseases and I think we're good. So botulism, it's bad in high doses. We use it in medicine, quote unquote, a little bit in low doses. What botulism is going to do is it's actually going to stop this interaction from happening. Stops muscle contraction. So all that acetylcholine that was going to break away out of here, botulism kind of turns that right around and says, nope. So putting injections of botulism in the right spot are going to mean that typical facial movements are not going to stretch and re-stretch those muscles as much anymore, and thus maybe they're going to last a little longer, let's say. Now, this is a bad deal if this is in a high dose somewhere, because as you can tell, if the muscle can't contract, sometimes you're going to lose that ability. So, can't contract, and then some of you might know this disease. Can't stop contracting. So normally, something called glycine and GABA, and again, you don't need to know these, but this is a good example of proof of principle here. Usually we have ways of shutting down the muscle contraction system. GABA is a neurotransmitter that does that. It calms you down, blah, blah, blah. It's a lot of stuff. Tetanus, unfortunately, stops the stopping mechanisms. It also stops acetylcholinease, right? So that acetylcholine is just flying around. The contraction stays semi-permanently, unfortunately, there. That's why when you have a tetanus patient, they're in a substantial amount of pain because their muscles are basically contracting all over the place. They have no control. Can you imagine the pain if your bicep and tricep were contracting at the same time? It gets pretty bad. And then eventually muscles in the back start to cause damage to the spine. So get your tetanus shot, to say the least. Okay, so I'll post these slides. This will have everything you'll need to know. I think these are helpful too. We also, this is kind of that cross bridge right here that I was talking about a little bit. Basically, all I need you to know for this stuff is that as this myosin head is going to interact with actin, each time it's going to bind, and then it's going to push. See how it does that, like bind and then push over to here at step 3. These 3 steps just repeat once it's unleashed. That's it. And you just need fresh ATP to keep doing this binding and push, unbind, and push, and that's it. So again, like I said, I'll post this. We should be okay. Okay. I think that we should be done. Sorry, I do a lot sometimes. But so for this one, like I said, you don't have to do any of the the reflex stuff. You can if you want to do the knee one, just to test it out because it's fun. But the main thing you're looking at is the EMG stuff. So let me know if I can help, and you're all set.